I would like to announce Ms. McInnes as our next presenter for Masterworks 2018.
Raise your hand if you know someone who's been affected by cancer. Cancer is the leading cause of death around the world, and its incidence continues to rise. Each year, 12.7 million people discover they have cancer, and 7.6 million people die from the disease. However, evidence suggests that 30 to 40% of these deaths could be prevented, and about 1/3 may be cured through early diagnosis and treatment. To put this number into perspective, each year in just North America, a population as large as Richmond dies as a result of cancer or cancer-related reasons. Hello, my name is Fiona McInnes, and welcome to my Masterworks presentation, entitled A Promising Future for Cancer Treatment, a study of the hallmarks of cancer and why nanomedicines can achieve better results in this complicated disease. Cancer is a huge topic, so I'm only going to cover a very small section of it in my presentation. I first became interested in cancer research last year during the Lower Mainland Regional Science Fair. I had the opportunity to go on tour of research labs at UBC, where technologies to detect cancer were being studied. I found the whole talk and presentation very interesting. This initially sparked my interest into cancer research. At the start of this 2017-18 school year, I already knew that I wanted to look further into this area of study, and so my Masterworks topic was decided. I would study cancer and cancer research. In school, I've always been interested in science, and it just so happens that at the start of the year, we were learning about cells, which led to learning about how cells divide and can make mistakes. I was intrigued by this and how this was even possible. When I first started my Masterworks research, I was overwhelmed by the amount of information I could find on the broad topic of cancer. But eventually, I narrowed my search down to one possible way of treating cancer: nanomedicine. Even this one topic is too broad to cover in a single thesis, especially for a 14-year-old. I realize that I've only studied the very tip of the cancer iceberg, but I hope to continue to learn about this subject in the future. Cancer is a devastating disease, and research is still needed to achieve better treatment outcomes. By the end of my presentation, I want you to understand 3 things. The first, what cancer is. The second, what the hallmarks of cancer are. And third, a little bit about nanomedicines. So first, I should describe cancer itself. Cancer is the name given to a collection of related diseases. In all types of cancer, some of the body's cells begin to divide uncontrollably without stopping and spread into the surrounding tissues. When cancer develops, the orderly process of cells dividing and growing is disrupted. As I was saying, as cells become more and more mutated, old, or damaged, some cells may survive when they should die, and new cells form when they are not needed. These extra cells can divide without stopping and can form growths called tumors. Most types of cancers form tumors, but there are some types of cancers in the blood that do not form a solid tumor. There are 2 types of tumors: malignant tumors, which are cancerous, and benign tumors, which are not. Malignant tumors spread and invade tissues in the rest of the body. Some cells from the original tumor will break off of the original area of growth and travel to distant places in the body through the blood or lymphatic system. Unlike malignant tumors, benign tumors do not spread or invade nearby tissues. Although benign tumors are not cancerous, they can sometimes grow to be quite large. When removed, they usually do not grow back, whereas malignant tumors often do. Now I'm going to talk about the differences between cancer cells and normal cells. There are many ways that cancer cells differ from normal cells, but here are 3 that I would like you to know about. The first is how they reproduce. The second is how they develop, and the third is called specialization and adhesion. The first and most significant difference has to do with how cancer cells multiply and reproduce. Cells divide for several reasons, including the need to form new tissue or to replace old and damaged cells. In a colony of healthy cells, this is a tightly regulated process. A bunch of chemical processes are needed to signal the cells when to divide and when to stop dividing. The difference with cancer cells is that they stop listening to the signals given to them by the body, and they divide uncontrollably without stopping. Cancer cells develop what is referred to as metabolic autonomy, which means they are able to self-mediate growth and division instead of being stimulated by an external signal like a normal cell. Another way cancer cells differ from normal cells is in the development process. Cancer cells are always present in the human body. However, they are typically recognized by the immune system and are destroyed before they can cause any problems. It is when cancer cells go unrecognized and begin to multiply that they become a problem. Cancer cells are unlike any other cells in the body, and they act totally out of line with normal cell functions. As they develop, they ignore the signal sent by the body telling them when to grow and divide and when to die. The third way cancer cells differ from normal cells is called specialization and adhesion. Before normal cells become specified cells, they are stem cells, made in bone marrow. The body trains them to do a specific job, like perform the function of a liver cell. And then the body sends them out to where they are best suited. The stem cell that is specialized to function as part of the liver will be sent to the liver, where it will adhere to similar tissue and assume its function. This is a fundamental process which allows the body to heal and develop specific tissue based on cell type. However, cancer stem cells demonstrate a different development process in which they never really become specialized and lack the adherence qualities that make normal cells stick with their own kind. Instead, cancer stem cells remain in the underdeveloped state where they continue to divide and spread. The second thing I want you to know by the end of my presentation is the hallmarks of cancer. But to understand this, first I need to talk a little bit more about gene mutation. As you know, cancer cells are normal cells gone whack, meaning they no longer respond to many of the signals that control cellular growth and death in normal cells. Cancer cells are thought to originate within tissue, and as they grow and divide, they diverge further from normalcy. During normal cell division, there are many checkpoints that look for such errors and either repair or terminate the cell. In the absence of these checkpoints, the cell can start to divide uncontrollably. But this alone is insufficient for cancer to kill. What causes checkpoints to be damaged are mutations. A gene mutation is a permanent alteration of the DNA sequence that makes up a gene, such that the sequence differs from what it should be. Mutations range in size. They can affect anywhere from a single DNA building block to a large segment of chromosomes that include multiple genes. To my right, you see a sequence of numbers, but where the numbers are in red, there's a mutation. There are two different types of gene mutations: hereditary mutations and acquired mutations. This will start our journey into what the scientists Douglas Hanahan and Robert Weinberg refer to as the hallmarks of cancer. To date, there are 10 hallmarks of cancer, which are features that define what cancer is and what it does. The hallmarks of cancer are the characteristics that all cancers have. They differentiate cancer cells from normal healthy cells. Scientists and clinicians use the hallmarks of cancer to be able to detect and diagnose cancer. I'm not going to talk about all the hallmarks of cancer in my presentation, but I am going to cover a few that I believe are important and interest me. The hallmarks of cancer can be thought of kind of like the monster that cancer is. Each hallmark differentiates cancer cells from normal cells. The first hallmark that I am going to talk about is genome instability. Not all cancer cells are equal. They vary, they compete, and the fittest survive to pass on their genes to daughter cells, which continue to vary, compete, and survive. If this sounds familiar, it's because it is. Cancer cells evolve. Within our body, cancer cells mutate and change and adapt to their environment. The evolving cancer cell has to outcompete the normal cells surrounding it, evade attack by immune cells, escape the apoptotic machinery which causes cells to self-destruct, corrupt and co-opt otherwise loyal surrounding cells, and migrate to distant parts of the body. In the picture above, every circle with a different color is a new mutation. Sometimes when genes mutate, those mutations are not corrected. It doesn't happen often. Each gene has only a 1 in 100 billion to a 1 in a billion chance of being miscopied. But each time a cell divides and passes a copy of its DNA on to its daughter cell, the chance of error increases. The average gene is home to somewhere between 100 million and 10 billion mutations spread across its copies in each cell. We know cancer occurs because we can see the stepwise accumulation of mutations. If a cancer cell acquired a mutation that enables it to grow faster or survive longer and produce more offspring than the surrounding cells without that mutation, then that cancer cell has a selective advantage. Its descendants will be fitter. They will outgrow and dominate the local tissue environment. You can think of cancer kind of like a monster. The monster is always having new problems thrown at it, and so it is always evolving to meet its basic needs and survive in its environment. Being a cancer cell means you must adapt quickly or die. If the monster doesn't evolve fast enough, then another cancer cell will come and take its place. So here I've painted three monsters, which you can think of like cancer tumors. So the two over there are cancer tumors that have just began to grow. But this one here has been growing for a long time and has developed more mutations that make it more adequate and better in the environment that it lives in. So if you're a cancer cell, think you have all these things that are trying to kill you, and the body doesn't want you, and there's going to be medicines and things thrown at you, and you have to try and survive. So if you're a cancer cell, which one here do you think would survive longer? The smaller one or the bigger one? Yeah, me too. So this is part of the reason why cancer tumors are always trying to develop so rapidly, because they want to be able to survive in their environment. Sustained angiogenesis. A growing tumor requires access to oxygen, nutrients, and waste disposal to thrive. The formation of new blood vessels, known as angiogenesis, satisfies these needs. Beyond a certain size, 1 millimeter, for reference, that's the size of the ball and the tip of the ballpoint pen, diffusion of nutrients and oxygen from the existing blood vessels is insufficient. The surface area to volume ratio becomes too low, and the developing tumor begins to starve. In response, cancer cells send out signals to the cells of nearby blood vessels, inducing these normal and healthy cells to grow extensions to form a supply chain and drainage channels. In healthy adults with no cancer, angiogenesis is only switched on during physiological processes such as wound healing or menstruation. In contrast, cancer cells always have angiogenesis switched on. Once again, going back to our analysis, once the tumor grows past a certain size, it cannot get enough nutrients into it by diffusion. So it tricks the healthy cells surrounding it into making secret tunnels called blood vessels that allow the tumor to steal nutrients that it needs to survive. The tumor also gets these healthy cells around it to make tunnels that take away the waste that the tumor creates. The last hallmark I'm going to talk about is activating metastasis. A growing tumor will eventually spawn pioneer cells. These move out of the original clump of mutant cells to evade adjacent tissues and then travel to distant sites where they form new colonies. These distant settlements of cancer cells are named metastasis. Metastasis is bad news and is associated with significantly reduced survival rates for patients and prognosis for patients. The ability to metastasize allows cancer cells to find new areas in the body where space and nutrients are not limiting. To my right, you see a cross-section of a human liver, taken at autopsy examination, showing multiple large pale tumor deposits. The tumor is an adenocarcinoma, derived from a primary lesion in the body of the pancreas. So, going back to our analogy of the monster, this would be the monster sending out children or minions to invade more tissues in the body. The minions are cells that have broken off from the original tumor. They find a suitable place to call home. At first, they have the same DNA as the original tumor, but because they're in a different environment, they have to adapt to these new conditions. So they will develop different mutations than their parent monster. original tumor that has grown very large, it's going to send off little cells from the sides of it, and these cells will go to invade other parts of the body. So you can think of this as the original tumor and these cells as little places that are going to find new environments in the rest of the body to inhabit. How cancer is currently treated. So currently, there are 3 main treatments for cancer: are surgery, chemotherapy, and radiation. Surgery is typically the first line of treatment if the tumor can be seen and removed safely. However, some tumors may have already metastasized, which means there are cells in the blood which cannot be removed by surgery. These cancers require different treatment or a combination of treatments. Even with surgery, there is a risk of local reoccurrence. Thus, most patients will have a combination of treatments. Surgery can be done in combination with radiation and chemotherapy. This is to ensure that all the cells are killed. Surgery. The oldest type of cancer treatment and still one of the most effective. Surgery is the removal of the tumor and the surrounding tissue during an operation. Surgery works best for solid tumors that are contained in one area. It is a localized treatment, meaning that it treats only the part of your body with the cancer. Surgery is often the first way patients are treated because it can be so effective. Surgery can be used for 3 reasons, depending on the cancer and how advanced it is. The first is to remove the entire tumor. Surgery can remove the cancer tumor if it's contained in one area. The second is to debulk the tumor. In this case, surgery removes some but not all of a cancer tumor. Debulking is used when removing the entire tumor might damage an organ or the body. Removing part of the tumor can help other treatments work better. And the third way is to ease cancer symptoms. Surgery can be used to remove tumors that are causing pain or pressure. The second treatment is chemotherapy, a type of treatment that includes medication or a combination of medications to treat cancer. This can be highly toxic, but new drugs are being developed to target specific elements that are only seen in cancer cells. The goal of chemotherapy is to stop or slow the growth of cancer cells. Chemotherapy is considered a systematic therapy, which means it can affect your entire body. Chemotherapy is designed to attack rapidly growing cancer cells, but they can also affect normal cells that grow rapidly. For example, the cells that generate blood cells, the cells lining the inside of your stomach, and the intestines, and hair follicles. The effect of these medications on normal cells is known to cause harsh side effects, like a reduction in white blood cells, hair loss, vomiting, and diarrhea, among others. And another common treatment is radiation. Radiation therapy uses high-energy radiation to shrink tumors and kill cancer cells in the area that is being irradiated. The radiation may be delivered by a machine outside the body. This is called external beam radiation, or it may come from radioactive material placed in the body or near the cancer cells. This is called internal radiation therapy. About half of all cancer patients receive some type of radiation therapy, sometime during the course of their treatment. Radiation therapy kills cancer cells by damaging their DNA. Cancer cells whose DNA is damaged beyond repair stop dividing or die. When the damaged cells die, they are broken down and eliminated by the body's natural processes. One of the issues with radiation therapy is that it also damages normal cells, leading to harsh side effects. A new way to treat cancer. The third thing I want you to understand at the end of my presentation is nanotechnology and nanomedicine. Nanotechnology is science, engineering, and technology conducted at the nanoscale, which is about 1 to 100 nanometers. Just how small is nano? In the International System of Units, the prefix nano means 1 billionth, or 10 to the power of -9. Therefore, 1 nanometer is a billionth of a meter. It's difficult to imagine just how small that is. So here are some examples to put this into perspective. On a comparative scale, if the diameter of a marble was 1 nanometer, then the diameter of Earth would be about 1 meter. A sheet of paper is 100,000 nanometers thick. A strand of human DNA is 2 nanometers in diameter, and a single gold atom is about 1/3 of a nanometer in diameter. 1 nanometer is as long as your fingernail grows in a second. Nanoscience and nanotechnology involve the ability to see and control individual atoms and molecules. Everything on Earth is made up of atoms. The food we eat, the clothes we wear, the buildings and houses we live in, and of course, cancer drugs. Nanomedicine is the application of nanomaterials or nanoparticles to medicine. Nanoparticles are a form of transport for drugs and can go places that drugs wouldn't be able to go on their own. Nanoparticles can be engineered and designed to packet and transport drugs directly to where they are needed. This targeted approach means that cancer cells cause the most harm in the particular and intended area of the tumor where they are delivered to. This minimalizes collateral damage to surrounding healthy tissues and therefore minimalizes the side effects. Drugs that are transformed into nano range offer some unique features which can lead to prolonged circulation, improved drug localization, enhanced drug efficiency. There are many types of nanomedicines that are already being used on patients, and many more that are in the developing stages. One type of nanomedicine is called an antibody drug conjugate. I spent a lot of time trying to research these complex technologies. Although there are many types of nanomedicines, this is the one that I found the most interesting to me. Antibody drug conjugates, or ADCs, are a new class of highly potent drugs. The antibody drug conjugate is a 3-component system, including a potent cytotoxic anti-cancer agent linked to a biodegradable linker to an antibody. The antibody binds to a specific marker at the surface of the cancer cell. The whole antibody drug conjugate is then internalized within the cancer cell, where the linker is degraded and the active drug is released. ADCs combine the unique, very sensitive targeting capabilities of antibodies, allowing discrimination between healthy and cancer tissues with the cell-killing ability of cytotoxic drugs. The key components of antibody drug conjugates include a monoclonal antibody, a stable linker, and a cytotoxic agent to target a variety of cancers. The cytotoxic anti-cancer drug is chemically linked to a monoclonal antibody that recognizes a specific tumor-associated antigen, making the drug combination very specific. The enhanced permeability effect is created when cancer tissues are formed. So cancer tumors have a need to grow at an uncontrollable rate and speed. But to do this, the tissues they form are formed fast and with very little care. Unlike healthy tissues that are created evenly with the drainage pathways, cancer tissues will be uneven and leaky, leaving gaps and holes big enough for antibody drug conjugates to get in. Tumor tissues usually lack effective lymphatic draining. So, in conclusion, I believe there are many possibilities in the near future that will change how cancer is treated, from detection and prevention to treating cancer itself. I have come to believe that using a combination of nanotechnologies is the most promising way to treat cancer, and that the hallmarks of cancer are a crucial part of cancer diagnostics. Cancer is a diverse disease, and there's still so much to be learned. With continued research and funding, I believe that the survival rate of cancer will continue to go up each year. I have learned that not all cancers can be easily cured, but that advances in technology and medicine offer hope. Medicine is a field of rapid advancement that has many obstacles to overcome, such as funding and regulations. My research of various cancer treatments has been eye-opening, and I look forward to new advancements and discoveries in treating the disease. So, my Masterworks wouldn't have been possible without my internal advisors, Pam Matthews and Anna Fenn. They were always available for meetings and to edit my paper. Their ideas and organization kept me motivated throughout this process. I'd also like to thank my parents for being supportive and providing food in times of mental crisis. And finally, I'd like to thank my external advisor, Dr. Marcel Bally. He took time out of his busy day to come to meetings, write emails, and edit my final paper. He also hosted Pam and I at the BC Cancer Agency, where we got the full tour of all the labs and input from experts on my project. Without all of you, there is no way I would have been able to accomplish what I have. Thank you for coming here today, and thank you to you for coming here today and listening to my Masterworks presentation.
Thank you for coming back for a very important announcement. And this is to say that Miss Fiona McInnes has met all the requirements of the Masterworks program at Island Pacific School.